One. Good morning. It's Monday. It's sunny. Well done, you. We're here, and we're going to continue our voyage into these invertebrate enthusiasms. I hope you had a great weekend. Thank you for the annotation. Don't lose your annotation tools, because we are going to be using them today. Clear drawings. All right. Let's go. So one of the first things I want to do is to say thank you to three of you, Hazel, Umair, and Natalie, who were three of the first people in to the invertebrate assignment, the invertebrate R, the invertebrateur assignment, and they did it to an invertebrate test kind of a way. They found that one of the packages, the designers of one of the packages, had made a major update on the 11th of September, just before your assignment went live on the 12th. And that update has an error in it. So if you've already been to the GitHub and downloaded, do it again. If you've done it anytime before 4 p.m. yesterday, so when you go to GitHub, you should see these last two, the R and the MD files, should be updated and fresh for you to see. So a round of applause and thanks all around there. And the fix is there. And this is a fix that you can apply yourself if you've already got the code. So I'd asked you to do is to go to last week was to go to, over the last 2 weeks, was to go to the Google Doc and tell me about your favorite invertebrates. And so I'm kind of giving the answer away here, but what is the red bar on this? So we've got predicted species diversity and named species diversity for all of the phyla. Annotate, you got text tools there, yes? Tell me about any patterns you see. So tell me about what that red bar is and any patterns that you see with it. So your annotate the tool will look like that. What do the red bars represent, and what kind of patterns do you see with them? Yep, the annotate tool right on the screen. That's where to write. You can write it in the chat too if you want, Kevin, but it can be here on the screen. Now I got some ideas. That's great. Awesome. Olivia, that's exactly it. It's the phylum to which your favorite species belongs to. So that is what the red bars represent. What kind of patterns do you see? The phylogenetic distribution of your favorite invertebrate. Any trends? That's it. Yeah, more and more common. That was the one that just appeared. More and more commonly known things. More preference for well-known species, I see in the chat, Olivia, as well. More diversity equals more favorites. Yeah, exactly. So I think this to me is one of the trends, is that even though we are a group of enthusiasts about invertebrates, our favorites still tend to track the things that we know the most about. And that's a bias of confirmation, or I suppose, or of familiarity. And it's a fine bias. Your favorite is what your favorite is. But I think the other thing to recognize is just all the love that needs to be spread, potentially, amongst all of these other clades here, where we have no favorites. And part of that has to do with the fact that we don't know much about them. We don't have many names, and these are the big differences between the described and the potential. So the described diversity in yellow and the potential diversity in blue. So lots of work to do. All right, let's take those away and we'll begin. So last time we were here, we were talking about the plankton. We were deeply immersed into talking about how, it's a hard knock life. It's hard to be a plankton, and different ways that plankton have of becoming food when you're very, very small and working in a low Reynolds number kind of environment. So we talked about all these different kinds of things, and we talked about emerging areas of plankton research. Today, we're going to talk about, and on Wednesday, this is a 2-lecture kind of a spread that is going to talk about life history. We'll talk about major traits affected and that drive life history evolution, like body size and lifespan. We'll talk about trade-offs, and we'll begin to talk about reproduction with kind of some shared terminology really at the end of today's lecture, and then we'll pick that up and spend more time there on Wednesday. So these are the outcomes. It's essentially entering into the concept of life history, how life history traits drive invertebrate characteristics, interpreting trade-offs, various examples of trade-offs, and various reproductive traits that are used by the vast diversity of invertebrates. Put in the chat, have you folks run into life history evolution or life history theory before in your ecology and or your evolution course? Yeah, great. Now, do you remember how many were you focused on invertebrate examples, vertebrate examples? Was it spread between the two? Both, I think. Vertebrate, definitely. Vertebrates. Yeah, and that's just another expression of our. So what we'll do then, as we go through, we'll have kind of a shared updating of lexicon, I suppose, to make sure we're all talking about the same things, contributing and thinking about the same pieces of pie when we think about life history theory and life history evolution. And then we'll dive into some of the invertebrate examples that having this 3700 course allows us to do. So life history, stripped of the theory, stripped of the evolution kind of suffix, you are basically talking about this pattern of growth and survival and reproductive that occur in the lifespan of the organism, right? So if we are looking, as we are here, at this life cycle of a freshwater mussel, of a unionid mussel, so this lineage of mollusk that would have hit its peak diversity in and around this part of the world prior to colonial settlement, where we've adults that are sticking their foot out into the substrate and releasing gametes into the water, and then glaucidia that go and search out like a little Pac-Man, and they go and they glom. Actually, I'm holding kind of a little Pac-Man. So they go and they glom on to the gills of the fish, and they suck the blood of the fish, and then they metamorphose, drop off, and grow larger and larger. So this period of basically of traits, this whole life history theory predicts how natural selection should parcel their resources into babies, which is one of my favorite quotes. This is a quote by Resnick that he wrote in 2010 in a book about natural, kind of where natural selection stands, particularly as it was expressed by Darwin. And the analogy and the reason that there was pi in that word find at the start is that is expanded upon by Resnick. So in our if we think about all of these traits associated with somatic and metabolism, somatic growth and metabolism, and traits associated with reproduction, they're all being drawn on in an energetic perspective from a pie, right? So this is the analogy of the pie, that you have a finite pie where maintaining your body is going to cost something, preparing your body for bad times is going to cost something, reproducing is going to cost something, and growing is going to cost something. And if one part of you is devoted to making babies to be larger, then that's going to take away resources from growth, maintenance, or fat storage. And this morning I was thinking about another kind of analogy that, if you haven't run into this theory before, that might make sense if you follow any professional sports league. You know that in the late 20th century and early 21st century, that most of the big professional leagues implemented a salary cap. So life history theory is essentially like a salary cap, where your Maple Leafs, my Montreal Canadiens, there's a fixed dollar value that team can pay its roster. And so the dollar that one player makes takes a dollar away from another player, as the great evolutionary thinker Elliot Friedman said in his 32 Thoughts podcast today. So this is expressed in the world of animals and plants, but particularly in invertebrates. And there's some classic expressions here, like with these crickets. And so crickets that have an intraspecific variability, so a polymorphism, so a defined phenotype. In this case, LWSW, which stands for long wing or short wing morphology in crickets. And so that's that polymorphism. It's in the same population. It's in the same species. And that these two morphs of the same population, the same species, they differ in a number of traits that are adapted to, in this case, dispersal, so flight, at the expense of egg production, and vice versa. And this is classically shown with a lot of gruesome cricket dissections, where in the long wing, when you dissect the thorax here, you can see the flight big, beefy, Schwarzeneggerian flight muscles with very small ovaries, and then big, beefy ovaries with very tiny muscles, very tiny flight muscles here. And my guess is that in your evolution and/or ecology introduction to life history, that you talked a lot about potentially in the application and maybe the history of RK selection, that the idea that there is a fast and a slow strategy to development, and that there is essentially kind of a teeter-totter that balances across in a community, and then within species within the community and populations within the species, where strategies can differ, where investments are either fixed as a slow strategy for development, for reproductive rate, or like that cricket, where they can have dispersal ability that is high or low, and some somatic maintenance that is high or low. So we've got here the idea that on the left-hand side, with high external mortality, that we see a lot of the driving effect of predation, of predators. And then on the slower strategy, we see the role of intraspecific competition playing making important contributions. And if you haven't been or if you like this, actually, so more so if you like it, I suppose, if you like this kind of catechus, or this roadmap to understanding how diversity and variation is separated amongst the world, and if you also are a fan of The Lord of the Rings, you will love this paper, which is all about how to teach life history evolution, or how to think about life history evolution using facts and characters from The Lord of the Rings. So what has life history? So this is Resnick wrote about when he was writing about, this is back in 2010, when he was writing about the impact of the role of natural selection in evolution as expressed by Darwin in The Origin of the Species. He talks about the fact that phrase is never used, life history evolution, but you can see its kind of precursor. You can see it kind of in the space between the lines. And really the expression of it, particularly the genetic and the mathematical expression of it, sprang into life about 60, 70 years ago in the mid 20th century. And since then it's been used to explain a lot of life history, like why some organisms are large and why some are small, to why they mature early or late, why they have more or fewer offspring, and why and when will they have a long or short lifespan. Body size is one of these traits that we use in life history evolution theory, and we use it a lot partially because it's correct. It's connected to a lot of these things, like it's listed there. So size at birth, size at maturity, patterns of growth, reproductive investment, which circles back to the size at birth, and environmental variables, all interacting with the body size of the organism. So these are some of the theoretical underpinnings of why body size is a really attractive life history variable. The other one is more pragmatic, and that is that it's something that we can measure really easily, generally, with most animals, that we can lay down, like with this rotifer, we can lay down into the microscope some kind of a scale to say this is this big, and we can even with large crabs, like on this gentleman's body here, we can measure body size even easier than we can measure body weight. So kind of it's two of the most simple organismal measures. Body size is kind of the biggest. And so let's talk about some of these life history theories associations as driven by body size together for a second here. So let's take this one. Find your annotate tool again. And let's say the scenario is we have on the x-axis the size of a host. And on the y-axis, we have the size of a parasite. What I'd like you to do is find your annotation tool and draw me what kind of a relationship. Say it's a continuous relationship. You can draw categorical if you want, but I'm imagining you drawing continuous. What kind of relationship would you expect to see between the body size of the host and the body size of the parasite? Awesome. That's awesome. That's great. Well done. So let me clear those for a second and I'll show you. Oh, competing hypothesis. So let's take a look at, so the two hypotheses, 2 to 3 maybe, I think I might have seen some asymptotes in there, but the idea being that the larger the host, the larger the parasite size might be. And you can picture that just in terms of, I think, the intuitive, that it's a positive relationship. You could express it just as that's the volume of the host is the world for the parasite, right? So the more world that there is, perhaps the larger you grow to expand that. So this is an example. This is actually an actual example from parasitic flatworms, from cestodes. And interestingly, so there is this positive relationship that you identified, and it's further driven by life history traits of the host. So there is a lot of driving of the parasite's life history by the host's life history attribute, the body size, but then a further one, whether it's endo or ectothermic. And so we have here the fact that you've got the same positive relationship. The slopes are roughly, at least according to a squint test, are about the same. And then, but the endotherm bodies, parasites, are going to be a little bit bigger again. Let's do another example. So we have echinoderm reproductive investments here. And this kind of ties back into some of what we were talking about last week. What I would like you to do this time is annotate on this figure, which has egg diameter on the vertical, and then different morphologies of echinoderm larva there on the right, and annotate which ones you estimate to be planktotrophic and which ones you think are lectotrophic. You do a P and an L with that text tool. Yeah. All right, I see. This is great. So one of the things I would point out here is that you've got these are little uh, yoke investments. This is great. Awesome. I'm going to clear those and I'm going to expose the rest of that. Fantastic. All right. So you've got here lectotrophic, leftotrophic, and then planktotrophic larvae. And I can add to this some pictures, some SEM pictures of what they would actually look like. And we're going to flip it upside down here. So we've got the scale bar down here. So from the bottom of the top, this is the brooded one. It's the legend that's flipped. So we're starting at the bottom with our lectotrophs here, and then comparatively, a much smaller planktotrophic larval stage there. And the scale bar down here at the bottom is half the size of the other. So this is a much larger investment, and it changes then the body size of these larval, as we've talked about in the planktonic or the planktonic kind of lectures. We've already invoked body size as being related to movement here as an investment. And so do you invest in a small number of lectotrophic eggs where each one of them has a greater investment because you're contributing yolk, and they're not good, so they're not going to move as far, but they are potentially hardier and going to be able to withstand kind of periods of low food availability. Whereas you can make a lot more of these, and they're potentially going to move a lot farther. So decisions made over evolutionary time, not with intent, just selected upon in the variability selected upon in the natural community, where differing investments have been made, in this case in movement versus reproduction. So life history traits can also affect not only body size, but lifespan and longevity. So senescence for organisms who reproduce sexually. So we see great variability where we've got red sea urchins at 200 years, other mussels at 400 years, fruit flies at 40 to 50 days. And then big changes in reproductive investment, of course, as well. So the life history traits can affect the number of offspring, like based on body size, based on growth rate, based on lifespan, based on age and maturity, based on predation. So we've got variation between millions of offspring per individual in the sea urchin to 10 offspring per brood in the Daphnia. Here's one example where these life history traits are varying based on, for these trichopterans. Now trichopterans, these little kind of, looks like a lepidoptera with kind of a tent-shaped set of wings at the back, and their larvae are aquatic, and they're, of course, you know these larvae, they're the ones that spin silk and gather substrate to make species-specific cases that protect the larvae and let them live. Now that silk costs energy. It takes it takes money for this NHL team to make their silk jerseys. And the silk production is really about defense, and it's costly. So there's a trade-off between larval silk production and the pattern of resource allocation that the adult can subsequently make. So what the researchers did was basically take controls and measure how much silk they were producing, and then they bothered and broke some of these defenses down to mimic predation, and made some of the trichopteran larvae rebuild. And so their investment, the male investment to silk, didn't double, but it went up significantly. Now the consequence of that, as an adult, those same adults were reduced in size. That by changing the amount of silk that the larva had to invest in, there was no longer a bank account that the adult could draw on to become as large. And this is the idea that there are intrinsic and extrinsic to the individual trade-offs that are happening. So trade-offs in physiological input that are described by developmental stage, that are contained within the genetic framework. But then they're also affected by the conditions and the factors outside of the individual. So environmental conditions, which can affect survival, age-specific rates of survival and mortality. Think for invertebrates about in our kind of an area about extrinsic factors might, if a particular life history stage might be the one that has adaptations for surviving winter, and if winter arrives early, and then downstream effects of that. So when we think of trade-offs with the pie analogy, that there's only, if we put a whole bunch, or the salary cap analogy, if we put a whole bunch, if your Leafs had put a whole bunch into Marner, they would have a lot less left over to spend on the rest of the team. And now if you're not going to invest in Marner, you'll see where you can make those investments. And so the more energy and time, in this case, in the x-axis, that it takes to make and create offspring means that the fewer offspring that it can produce. This is essentially an expression of that echinoderm larval graph that you were annotating earlier on. So let's look at it in terms of metabolism, though. Say I'm interested in you now, annotating on this figure what kind of relationship you would expect with energy into reproduction versus energy into growth. So a graphical expression, essentially, of this of the trade-offs that are inherent to life history theory. There we go. That's awesome. And I see some competition or some diversity, that there's a big amount of you just thinking about a linear relationship, and that's great, and some with more of, I guess, an upside down L or a U-shaped, also great. Both can happen in the natural, I could imagine conditions where each of those would happen, but the majority of what we would expect to see, let me clear these thank you, is that which the majority of your lines were. So neither was wrong, but this is the majority of cases that we would expect to see, right? Because energy spent growing is energy that can't be put towards reproduction. So this is kind of an interesting example where there are females laying eggs, and there's two behaviors that are expressed by the females. They can either guard those eggs, they can provide provisions for the eggs, or they can just lay them and move on. And I'll add some color here to show some of the differences. So the non-carers, essentially, and it's the top and the bottom category. So this is, imagine the garters as kind of an intermediate, but we can see that for the non-carers, their lives, because they're that trade-off means that they can just get bigger and bigger, and their fecundity still tends to increase with size. But if you're going to care for the eggs, then larger females declined in fecundity. So their lifetime productivity was reduced. So when we think about, whether it's body size, whether it's reproductive investment, and I mentioned when we talked about body size earlier, this isn't just an esoteric part of your degree at Guelph. This is a press release about a paper that came out this summer about the release of a large marine database, hitherto unseen database that looked at body size and tried to compile estimates of body size for as many species in the marine environment as possible. This is so the Marine Organismal Body Size Database, or MOBS. So they include the maximum linear dimensions of marine animals, height, length, width, and diameter. And so we've got the world has now, as this database exists and it's going to be added to through time, for all of these species. And you can see the account, but the estimate of kind of studies that are included in this database by thinking about all of these different categories and looking at the count size there. That there's in thousands and thousands of studies in this database, thousands and thousands of estimates. So, looking at this frequency histogram, that's of occurrence of species, of size of animal in the marine environment. How would you answer the question, are most of the species in the ocean large or small Reynolds numbers animals? And remembering that that's about where 1 millimeter would occur in the logging of the length that is, in this case, a centimeter scale. So put your vote in the box. That's it. That's totally right. So most are large. So most of the animals are on this end of the distribution, okay? But I think it's two things. One of them to consider is there's a whole large amount of species that are under that. And then I would say, erase your answer for a second. Actually, I'll do it. It's quicker because I'm going to change the question slightly. And say, okay, I agree. Most of the animals, most of the species are large. Now imagine that said most of the individuals. So irrespective of what this says about diversity, where are most of the individual animals within and between all of these species? Yeah, that's it. So most are living in that really viscous world, that low or small Reynolds number world. And although most of the diversity is larger, a surprisingly large component. And I emphasize this only because our intuition about what we bring to the table in terms of seeing, and in this case I'm using seeing as a synonym for understanding, how these species are living and experiencing the planet, is good over here, generally. If things are big, like us, in the same kind of approximate size range, we're pretty good at understanding. When they're small, we're less good. Okay? That's where we have to think where common sense doesn't make sense, because our common sense is inside of this large inertial frame. And so when we're very, very small and often don't have a name, that's going to be a problem. Our intuition is not going to be as helpful. I'm going to ask you a different question about, so that's this same database, the MOBS database, now thrown onto the Dunn et al. Phylogeny, again with the log average body size. So let's say, and we'll apply that Reynolds number right about there, and say, are colonial animals living in a large or small Reynolds environment? And so let's say most of the colonial animals are in these three phyla. Large or small? I suppose you could put a vote on the top for large of the name of the box, and on the bottom for small. Yeah, sponges, most sponges are big things. Most cnidarians, colonial cnidarians, they're big things. Let's see, we'll kind of move from the bottom of that screen into within, and look for bryozoans and say, ooh, lots of variability, but if that red bar indicates the mean, they're smaller, but they're kind of both. So this is interesting, right? This is thousands of studies, thousands of pieces of information that let you parameterize your future hypotheses about colonial living, about rates of reproduction, about dispersal probabilities. And let me thank you for that. Let's clear all that. And Yes. So the next thing. Oh yeah. Hi, Caramel. We haven't seen you in a while. So plankton trophic are long-lived. Yes, that's correct. Because they're feeding. Shout out to freshwater mussels. Okay. I'm scanning quickly through. I don't see any. I think I saw them all there. Let's switch gears for the next, say, 10 minutes or so and talk about reproduction. With some statements that make sense. Reproduction is critical. Survival, your survival from an evolutionary perspective is relevant only as it affects reproduction. That's a very strict kind of Darwinian way of thinking about things. And so but it begins us, or it lets us then think about the unit of selection as being the individual, the organism, and sometimes the relatives. So let's take a look. We've talked about some of the cnidarians, and so we're looking at reproductive investment in some corals here, where we have gametes being shed out into the water. The lunar conditions are the correct, and so we see this kind of filling of the water column with male and female gametes that bump into each other, and we see sperm accepted by the egg, and then developmental stages, and all of this happening out freely in the ocean. Then for that particular cnidarian species, a planula larva, which looks a little bit like, to a lot of people, like a little placozoan, beetling around, very simple amoeboid looking metazoan that then falls out of the water column, and as it falls out of the water column and settles eventually, it begins to change its form and shape. It tends to orient itself with a base that attaches, and then a body that begins to assume and take in its symbionts, the dinoflagellates, the zooncell and antheli, and now we know these apicomplexan symbionts as well. So, hooray, hooray, hooray for this kind of small episode of life history of a particular cnidarian. Now, when we think about the reproduction within animals, within the most interesting and diverse animals, within the invertebrates, we're thinking about asexual, sexual, and then some combination of the two. And we spent a lot of time as we work through the Dunn et al. Phylogeny in 2700 breaking down specific examples. And so the next few minutes are really just a reminder of some of these terms and of some of these examples that we'll then dig into more as they pertain to life history theory and life history evolution on Wednesday. So when looking at the distribution or the adoption of different reproductive types or strategies, the things to consider that we would have to do is how many offspring to produce, how much to invest in each offspring, and how often to reproduce. All thinking back to these reproductive decisions being made not intentionally by the organism, but by the environment upon the organism. So, when we think about asexual reproduction, there's some classic invertebrate examples, right, with facultative parthenogenesis that we see in the Daphnia, and the obligate asexuals like some of the rotifer lineages, and they reproduce either by subdividing an existing body through budding or fission, or the production of diploid eggs, so parthenogenesis. That can be facultative, so opportunistic responding to potentially, in a lot of lineages, to poor environmental conditions, or obligate. And it can be a really, or good environmental conditions. There are examples of both, but it's a very efficient form of reproduction. When we think of asexual reproduction via fission, it's common in a lot of the soft-bodied phyla. So think about that. So we've got sponges and cnidarians in the Dunn et al. Phylogeny. Then we've got platyhelminthes, nemertean worms, annelids, some echinoderms. We see examples. So is fission a monophyletically distributed trait? It is not. We don't see it often in organisms that have hard external casings. So arthropods and mollusks, some of the most diverse, that is species level lineages, we don't see a lot of fission. And this can be a simple transection into two fragments that can give rise to one or two multiple fragments. So you you have raised with Sherry last year, and spoiler alert, I think they pull this away, but this is of course looking at the fragmentation of the flatworm, of the helminth flatworm, and the Nobel Prizes that have been learned and given through time to developmental biologists that figured out the number of times that this can happen and the developmental processes that take place. So we see here the healing with the head and those optical receptors at the front, and then a mid part of the body. And if we keep going along, we can see overnight big changes. And then in this middle section, you see those two little darkenings there. That's the development, essentially the reorientation of cephalization of the head of these eye spots. And then over a period of days and weeks, we end up with multiple individuals that have come from the, in this case, traumatic transection of a single. Incomplete fission can give rise to colonial organisms, such as some bryozoans, some cnidarians, and even some urochordates, some deuterostomes. So, like our part of the phylogeny of animals. That tends to make the concept of the individual. So, at what level is selection acting on? Is it acting upon the colony? Is it acting upon and so if we can't at a course level interpret that, if they're made up of modular units as opposed to individuals. So being that if those modular units are not affected as individuals by whatever selective event is happening, then it's going to be most efficient to model the individual as the colony, not as the component. So asexual reproduction that involves the use of eggs. We have cases, obligate parthenogenesis, where we spent some time in 2700 talking about that with bedolid rotifers. And we would see parthenogenesis rolled out more occasionally in other lineages cyclically, where there's diploid eggs that develop into new individuals, and then occasionally there's sex that happens, and then they're sexually reproduced individuals. And then we have cases like this where there's arhenataki, where unfertilized haploid eggs develop into males and fertilized diploid eggs develop into females. So we see in these Daphnia where we've got a parthenogenic lifestyle, so the female produces a parthenogenic daughter, and a parthenogenic son, that kind of at a lower frequency. But then we've got a haploid egg, and that can become involved in the sexual cycle with whether it's a male or a female out with those floating low investment males. Then we've got sexual eggs that hatch after dipause, tending to become females. And so this is a strategy that tends to deal well with that variable environment that I invoked earlier. So a drought-prone environment or a winter-prone environment. And it's a common strategy in the marine environment. So this alternation of sexual and asexual reproduction is really common. Think about the classic hydroid here. So we've got the hydroid colony on the left-hand side, the large thing that is going to bud off, and we've got a young hydroid colony. Now, it's been formed, I'm working backwards here through, but that planula larva that we saw moving around formed because we've got eggs out in and sperm out in the water column. So we've got medusoid and hydroid forms. We've got budding and then a sexual stage as well. So there's the asexual polyp of Obelia here, budding off, and then we've got the sexual medusoid form that's formed out in the water column. And that, of course, is a cue for me to show one of my favorite invertebrate little gifts, which is this strobulating Obelia. So these little medusoids kind of budding off of their original colony, strobulating out and then releasing the gametes. And so we've got here the classic Medusozoan cycle, and then the Anthozoan, the corals, do essentially this part. And that's back into 2700. So that is the extent of where I wanted us to get today, because I don't want to pull in too much all at one time. So we've talked today about kind of a shared introduction to life history, a reintroduction to life history and the trade-offs that an organism makes with respect to time and energy and biological processes. We kind of then worked our way into some of the classic invertebrate examples of different kinds of sexual reproduction. And then next time, on Wednesday, we're going to talk about a lot of life history theory as it affects reproduction, male and female investment in reproduction. So what else should be going on in your life according to this class right now? You've got the Invertebrate R assignment this month. It's open and available and working for you to work on. There'll be case studies, and there's my encouragement to form kind of study groups, 3, 4, 5 person study groups, to make yourself questions, to prepare for those case studies. And you've got what looks like a beautiful day outside for your lab in the Arboretum. And if you haven't already, check course link to make sure today, check course link for sure, to be sure. But today looks like it's going to go ahead, and then in the coming days, just check course link before you go out. And then course link also has a copy of this map that tells you where to meet. You'll need a piece of paper and something to write with, and that's about it. So take care of yourself, take care of others around you, and enjoy your time in the Arboretum. And we will rejoin each other on Wednesday to talk more about life history theory as pertaining to invertebrates. So I'm going to stop recording, and